All right, thanks a lot to all the organizers. This has been fun and I look forward. I think this is a very interesting meeting because it allows you to sort of ask interesting questions which have not been solved. And this is what I'm going to talk to you about a little bit today, about this notion of single cell states and whether they are always very meaningful in terms of the underlying biology that you're trying to learn. Okay. So of course, I mean, to this audience, I don't have to introduce this, but since single cell technologies have become more easily accessible and cheaper, there's a big general question about identifying states at the single cell level and being able to connect it to ultimate fates, right? And we've looked at a few examples of this before. But something that's been, I think, a challenge across different fields is to sort of really define what do you mean by a state a priori, okay? And often what I find, at least I found it quite challenging when I read papers, to sort of figure out whether it's a circular definition. Often I see that cell states are defined by dimensionality reduction, and then you actually don't know what was the original definition. What is it that you're finding? Of course, if you do dimensionality reduction, some similarity metrics, you will always get a cell state. But is that really meaningful? And so today what I want to talk to you about is an interest in our lab, which is on the circadian clock. We are interested in it for multiple different reasons, but I thought it would be a nice thing to present here today because this is a system where I feel the notion of a cell state is very well defined. This is well defined before you try to do anything else, right? And then the question is, if you have single cell level data, can you actually decode these single cell states, okay? And sort of in keeping with the spirit of this workshop, I want to be a bit provocative and tell you that in certain circumstances, we feel that making state assignments at the single cell level using RNA is completely meaningless, okay? And I'll try to tell you why we think so. So because this system may not be familiar to a lot of you, let me first give you a very quick overview of what the mammalian circadian clock is. You basically have a set of gene regulatory networks. There's a negative feedback, there's a time delay, etc. A transcriptional translational feedback loop that generates these oscillations. And ultimately that reflects in the oscillations of a bunch of RNA and proteins, okay? Now, the important thing to remember here is that this oscillator is about 24 hours, not exactly 24 hours. And then when you interact with the external day-night cycle, you get entrained to this day-night cycle and you get 24-hour rhythms, okay? But there is this notion of a phase of entrainment, which means if I were to take, say, a single sample of, say, skin from each one of you in this room, when we are exactly at 11 o'clock external time, what will happen is we will all be spread out on the circle, okay? So the notion of a cell state is where you lie on this circle, okay? So this is an oscillatory system. There's a periodicity once you've been entrained, and the phase of entrainment is the state of your circadian clock, which can be represented as an angular variable on this circle, okay? So the definition of a cell state is very clear. It's where you lie on this circle. And the question that you can ask is, if you have these gene expression data sets from a snapshot, so for example, if I take gene expression from snapshots from, let's say, skin from all of us, can I tell you from that gene expression where you are, okay? So can I decode the state of your circadian clock? And what is very obvious, right, is that to be able to do such an inference, you will need at least 2 genes which are slightly phase shifted from each other, because otherwise with just one gene, you cannot do this. Your y-axis will have multiple phases where you have the same values, okay? So at least 2 genes is what is theoretically required. But then the question is, of course, it's much more difficult than this. And the questions that we are interested in the lab are the following, right? In this particular system, this is quite complicated because of low copy number noise. I'm not sure what happened here. Okay, yeah. So in principle, 2 genes should be sufficient, but is it really sufficient? And the answer is, of course, no, because this system is very nice because in terms of the challenge that it poses, because typically these genes express only about 10 to 20 RNA molecules per gene. Okay? Compare this to the cell cycle, which has been much better studied, a similar kind of question, but in the cell cycle, typically you get hundreds of molecules. Okay? The second challenge is you have very few oscillatory genes across conditions. Okay? So this inference problem that we are trying to solve is using of the order of 10 core clock genes. Again, compared to the cell cycle, you have about hundreds of such genes. Here you have only about 5 core clock genes. Okay? And so the broad question is, when you have such few genes to work with that are physiologically relevant, this notion of a single cell circadian clock state, is it even meaningful to ask, okay, with such low numbers of genes when there is so much expression noise? Yeah.
So are there cases where you have like 2 oscillators running, and is that a problem? If you injure the liver, will you have hepatocytes that re-enter the cell cycle but are also trying to oscillate in a circadian manner? Okay,
so this maybe we should have this discussion. It is a very good question. Yes, there are various hierarchies of oscillators in the body. And what you're saying is right. If one gets damaged, the other might still keep running. But the point is, as long as your circadian clock is intact and it is entrained to the day-night cycle, we have a well-defined problem where we have a 24-hour oscillator. And from the genes, you should be able to infer phase. Okay? The third important challenge, and I'm going to show you this in reconstructing phases from single cells, etc., is the fact that you have phase dispersion within single samples. Okay, so even if your system is very well entrained, a single sample will typically have phase dispersion. Okay, so you will have multiple cellular groups coming from slightly different cell phases. So with this sort of a background, the kind of questions that we are currently interested in the lab are the following. Are there fundamental limits, not just technical limits, okay? Because remember, if you have a system where there is so much noise, what will immediately happen is when you're looking at data sets like single-cell RNA-seq, for example, the issues of sequencing dropouts, etc., become particularly exacerbated. But with all of this, are you still able to say something about more fundamental limits that are set by biology and not the technology? Okay, so these are the kinds of questions we are asking. Are we able to actually infer phase from single cells? When you can get rid of these technical issues? How do we do this? Can we do this from single cells? And of course, because now you have single cell data, an interesting question that you want to answer is, are you able to extract desynchrony from these kinds of samples? Okay. So these are the broad kinds of questions that we are trying to answer. Yeah.
Biology, you gave an example of skin, but he gave an example of liver, which I think may care more about circadian cycle. Does it depend? Like, do you really want to look at day-night cycle in skin cells?
I mean, yes. I mean, there are many reasons why you might want to look at day-night cycles in the skin, and we can talk about that later. And the idea is, as long as you have this small set of genes that you can reliably measure, it doesn't matter in terms of the underlying concepts that I'm going to discuss, whether you're looking at liver or skin, etc. Okay. All right. So the first question we asked is, because these are very noisy genes, can we figure out if technical noise is going to create issues, or if the real underlying gene expression noise is going to create issues? Okay. So what we did was we set up an experiment. These are the sort of more modern age technologies that allow you to do imaging of many genes at the same time. This is saberfish. It was developed in the Wyss Institute. And what we did with this technique is we measured up to 8 of these core clock genes in hundreds of single cells. Okay, and what we could show is that these technologies give you about 95% measurement accuracy. Okay, which means that out of 100 genes, 100 RNA, we are able to capture 95 of them. Only 5% is dropout. Okay, so this has allowed us to take care of one major technical issue, which is the sequencing dropouts. Okay, with this, what we did was we designed a very simple experiment where we took fibroblast cells, synchronized them, and then let them run for different time intervals. Okay, so these are different samples, all started off in a completely synchronized state. Then every 4 hours, we are taking a different sample, freezing or fixing them, and doing this kind of multiplex saberfish measurements. Okay, so for each single cell, we have measurements of between 4 to 8 genes of the core circadian clock. Yeah.
How do you synchronize the cells? Because you mentioned that if they just entrain, then they're not necessarily synchronized.
Yeah. So let me get to the next slide. So that's my next slide. Okay. So you're right. So dexamethasone is supposed to be a very strong synchronizing agent, but there are issues with the synchronization, and I'll tell you about that in the next slide. Okay. And the question we first asked is, in this setting, can we actually infer the underlying true phase of these single cells? And remember, because this is a dexamethasone synchronized setup, each of these hours, these are our Zeitgeber times. So we sort of know the ground truth of these experiments. Okay. And so now what we are doing is, from the gene expression measurements, we are trying to ask whether we can actually tell if this single cell came from ZT6 or ZT10 and so on. Okay. So that's the question we asked. And here is the plot. This is on the x-axis telling you how many cells we've averaged or pseudo-bulked over. This is real single cells. And on the y-axis are the errors from our phase inference. Okay, so we have different methods. We tried multiple different methods of state inference. We know the ground truth, so we know the errors in our phase measurements. And what you can see very interestingly is, at the single cell level, our errors are essentially spanning a complete 12 hours. Okay, what this is telling us is that the errors in our measurement are so large that we essentially are just sort of guessing at random. Okay, and interestingly, when you start pseudo bulking over samples within these ZTs, you start seeing that you can get very reliable estimates of the underlying ZT that these cells came from. Okay, so your errors now go below half an hour. So coming back to your question, there is a problem, there is a limitation of these experiments, and that's exactly what he was pointing out. There is some degree of desynchronization happening even in these samples. Okay, this is something that you simply cannot overcome, and this is a very technically challenging thing to overcome. And so the next question we asked is there any desynchronization really happening? And the answer is yes. So these are time courses of these genes, and you can very clearly see an amplitude reduction in these oscillations, telling us that there is indeed some desynchronization happening. And if this is really happening, this is a problem, because this will then increase the dispersion of gene expression. And maybe that is why we were not able to infer the real underlying single cell states in the last slide that I showed you. So to overcome this, we designed a very simple experiment where we decided not to add any dexamethasone. Okay, so the whole issue of desynchronization is coming because we are trying to synchronize these cells, and some of them stay unsynchronized. So this is an experiment we did where all the cells are completely asynchronized. Okay, and what we are now doing is we are also taking into account the spatial location of these cells. Okay, so the entire dish is asynchronized, but what we do know from other protein reporter studies is that there are local regions of synchronization even within this totally asynchronous population. Okay, and the question we asked is, if we do this kind of phase inference from the genes, the gene expression at single cell level, can we pick up these spatial patterns of synchrony even in the entirely asynchronous population? And interestingly, the answer was no. So this is the result. So these are individual fields of views that are separated in space. And if you try to use single cells, what you see is that your phase is basically all over the circle for every single field of view. Only when we did some kind of local spatial pseudo-bulking were we able to find the well-established result that in space, you should be able to see regions of spatial synchrony. Okay, so each of these colors represent a spatial group, and these arrows basically is what is called the Rayleigh statistic. The longer the arrow, the more tightly synchronized these are. You can see that the entire circle, the Rayleigh statistic is very small. The smaller the number, the more dispersed it is. So this makes sense because it's an asynchronous population, but you can see that within the spatial groups, you have a very high Rayleigh statistic. And this we can discover only when we are doing the pseudo-bulking. Okay, so I want to pause here for just a second to let you think about this. What we are now saying is that in a setup where the definition of the cell state is very clear, and we made that definition explicit to begin with, right? We are not sort of doing some kind of dimensionality reduction to discover cell states. We know these cell states. We are using single cell gene expression to try and recover them. We are finding that it is impossible to do that with these number of genes. We've done it with up to 8 genes. And if you try to do it, you are actually not able to recover the underlying biology that exists. Okay, so we think that this means that if there is a lot of noise in gene expression, which is really the case in these kinds of circadian clocks, you have to be really careful when you're trying to discover single cell states. Okay, it may be the case that you're really just discovering the effects of noise. So I think his hand was up. Yeah, I mean,
there must be computational models, at least partially, of your circuit.
Yeah.
And you could extend those to be stochastic.
Yes.
Models.
Mm-hmm.
Is there a consistency that if you go and look at the level of noise you see in a typical model, given the number of proteins that you said, that it makes sense?
Yeah, so we've done all of those. In fact, those are published, where you take these models of the circadian clock, you put in the kind of measured noise that we have, and you see that you will just simply not be able to do it if you have this level, this number of genes. Okay, and so again,
So perhaps I'm missing something, but is this inherent to the lack of sensitivity because of mRNA measurements? Or
Yes.
So if you imagined a case where you actually had the ultimate degradation measurement, right? Could you actually properly place these cells along the oscillatory?
So good question. So we think that this is a limitation of looking at just RNA. Like Julie was saying, it's not just RNA that defines your cell state, right? It's a much more high dimensional quantity. If we, for example, also had protein measurements, it's very likely that we would have been able to place them correctly.
This is very specifically a coupled transcription translational
Yes,
feedback system.
that's right. Yes. But here we are only using RNA, right? Because that's typically what a lot of people want to do, use RNA to infer cell state.
If this is a very robust mechanism, then it shouldn't make too much difference what you use as a probe to see that effect, right? It's basically with RNA, but you could use something else. Not that this is the only important thing, but it has enough information for you to see the phenomenon you are trying to look at.
Right.
And also, this has to be a robust process. So it's hard to believe that depend on very precise numbers. Otherwise, biology won't work for this case. So how do you reconcile all that?
Well, I mean, it's possible that, the information is encoded in many more genes, perhaps, or in the protein levels. We know, for example, in the circadian clock that proteins are expressed in the order of thousands of molecules. So potentially it's encoded much more in proteins. I'm just saying that if you are stuck to measuring only RNA, then maybe it's a bad idea to be assigning state just based on, say, some marker genes where we know what the underlying phase really is. Okay, so maybe I'll just quickly, because there's only 2 more minutes, I'll just quickly go through the rest of the things. So basically what we think is the better way to approach this phase inference problem, and this also actually has relevance to inferring in the cell cycle, is to actually do a little bit of pseudobulking. And so what we've now developed is a way in which you don't have to pseudobulk over hundreds and hundreds of cells, which is typically what is done. We have a way in which we've developed a variational autoencoder. This hopefully should come out very soon, where you can sort of coax your cells to get tighter on the circle by some clever manipulations, which allow you to do sort of write down these hierarchical probabilistic models and then sort of force each of your cluster to become tighter. I'm happy to talk about this. It's a bit technical, but with this, we asked a simple question. I'm going to skip all these, but I'm just going to leave you with this result where we basically then asked, are we able to figure out how much desynchrony there is in these samples? Okay, and what's the evidence that we can actually get out sensible notions of desynchrony? So remember the experiment I was talking about where we have these time course gene expression measurements which are decaying with time. So this is at the gene level, right? And what we can sort of do some very simple calculations from oscillator theory to show that these curves will basically decay with a function that looks like this, where this sigma basically tells you how much spread there is in the frequencies of every single cell in your system, okay? And that spread in frequencies will give rise to this decaying function. So we fitted this with this function and extracted the sigma out of it. So this is about 0.02 per hour. And then we asked, can we actually get these numbers out from our single cell analysis, right? And again, what we found is only after we do this kind of pseudobulking, and we use this method that I showed you before, this decipher method, we can calculate the circular standard deviation from each of these ZT, these time points, and we can see that the slope of this line, which is expected to go as sigma t, and this gives you an independent measure of sigma, sort of nicely matches with the kind of slopes that we had from the gene expression measurements, okay? So suggesting that from these kinds of single cell measurements, if you're careful and you're not doing it at the single cell level, but rather at smaller pseudobulk levels, you are actually able to nicely extract the phase and also the kind of phase desynchrony from these measurements, okay? And you can do this with single cell RNA-seq data, but I'm going to just stop here and summarize and sort of leave you with this thought once again, that when you know the real definition of your underlying state, it may or may not turn out that your inference of these states actually gives rise to the real state that you're looking for. And this is one thing we are seeing with the circadian clock. I don't know how broader or more general this will be, but interesting thing to keep in mind is the median level of gene expression of most biological systems is actually of the order of 20 to 30 RNA molecules, which is exactly like that of the clock, okay? So if this is affecting the clock, my sense is this is going to affect other systems as well, okay? And maybe I'll just stop here.
Can I ask my question again? Yeah. You're telling me based biologists don't know anything about pseudobulking. So this pseudobulking, you say you're doing that where you only look at RNA, or what's the biological connection here? And actually the information is somewhere else. So how do I reconcile all that? Yeah,
so I mean, the way I'm thinking about it right now is that maybe this phase information is better stored in the protein molecules. But the problem is that measuring proteins with, say,
I understand that, but why pseudobulking solve my problem?
That's because if I can show you the figures. The reason pseudobulking is solving you the problem is because at single cell level, the gene expression distributions are very wide. So at different ZTs, your distributions are completely overlapping. But if you now start pseudobulking over cells within these ZTs, you're basically shrinking the variance of these distributions.
I understand that, but if that's the case, then the information is there. If you're able to take the information is there, you don't have to go to the protein. So that's where I get confused. There's a circular.
Yeah, so I mean, but the question is, how do you extract that information, right?
That's a problem we all have.
Yeah. So maybe the information is there.
But on the protein.
Yeah, exactly. Yeah, I mean, I don't know. I mean, the point is that information might be there, but is it there in the averages? Is it there at the single cell level? Yeah, I mean.
So pretty cool.
I just follow that very quickly. Bring up an important question. Many people who not familiar with single cell analysis don't know that you should never use raw data. You know, this was a really new thing for single cell data. First of all, high throughput. People like high throughput, right? It's very lot of noise, but high throughput, high dimensional. So you should process data. You know, when you do this bulk average, you do right. You have data manifold. You do local average. That's right. In cell, to answer Jose's question, cell may using this time average. Right here, you see a local example.
Spatial average.
Okay. Yeah. Another thing, high dimensionality. You know, that related to Leo's talk about, right? So even you have a lot of gene drop out, because you have high dimension, you have those one in the middle, that will help you to recover those drop out genes. You should really process first.
Oh, okay.
That is, you know, many people, if you do not really touch on single cell data carefully, you don't know that.
All right. Okay. Let's thank Shaon. And
No.